I think we're live now, but I will still take a couple of minutes before we start, Naren. It's 11:29, and I've learned that Indian stretchable time still exists on webinars, so you'll see 11:31 more people coming in.
All right, no worries. And I think people may be coming out of the Prime Minister's talk.
That's right. So it's May 3. So what do you think? It's going to be extended beyond May 3, or May 3 is final?
Well, I think the longer any economy can sustain to be socially distant, the better it is, because it gives the possibility that people who have been infected or exposed can clear the virus. Fortunately, for the people who are healthy, the virus is cleared within a month. That's what we believe today. So if we give a month for people, or at least 2 months for people to get rid of the virus, then it'll be better for society. So I don't know. We'll see. We'll have to data. It's all about the data and how the data comes in.
Fantastic. I think it's time now, so we'll go live. So good morning, friends. Once again, welcome to the 5th episode of the Yogananda Guru series of webinars powered by Shoolini University. We are all suffering from COVID. We're learning to live a new life. But we're all intrigued with a very simple question, and that simple question might be very complicated, and that is, what is this whole COVID all about? What is this virus all about? Why is it so different from influenza and SARS, for example? What makes it so virulent? When is a vaccine coming? When can we get out of our houses? When will life be normal? Of course, there is a social distancing angle to it, but then there is a science behind it. There is a biology behind it. And we are delighted to welcome today one of the world's leading biologists, biotechnologists, Narendra Chirmule. Quickly, Narendra is a dear friend. He's also a visiting faculty at Shoolini University. But more importantly, he's had multiple positions in great firms like Amgen, and his last formal role was head of research for Biocon. He now runs his own biology-centric data analytics company. Naren is actually in the US right now, in Philadelphia, if I'm not wrong, and it's 2 a.m. for him. So we can see, I think he's got the right background also. It looks a little dark and dusky out there as far as the background is concerned. And we can also see your fan revolving, so it must be a little warm out there, Naren. But anyway.
Inside, yes.
Yes, of course. So I think with that introduction, once again, welcome, Naren. Welcome everyone on this webinar. I think it's going to be a fascinating discussion. We have one of the world's best people to talk about the biology and the science behind COVID. Naren will take you through this fascinating journey, but he'll speak in English. What I mean by that is he'll try to explain the science of COVID in layman terms. So before we actually get into this, Naren, I have a very simple question for you. What made you get into becoming a scientist? What made you move into biology? I think we'd love to hear about that before you get into your lecture.
Yeah, good question. Thank you. First of all, Atul, thank you very much for inviting me to this talk. I enjoy all my visits to Shoolini University. It's an exquisite university, young, vibrant, and Dr. Khosla and you and all the team members in the university are so enthusiastic. I love being there. I wish you all the success in your goals that you have behind you. So my journey into biology was a little serendipity, and one thing leads to another as we are growing up, and you get inspired by people that you see and mentors that you follow. So whether it was in school, Dr. Ganguly, Dr. Gon, Dr. all of these teachers really influence you to decide what you're going to do in life. And then Dr. Bhagwat in Junjunwala College in Bombay, Dr. Dev, Dr. Mulherkar in Cancer Research Institute. I can go on naming people, but sort of people who inspire you lead you to your journey. So that's sort of been my story.
Fantastic. So I think we're all eager to hear from you. So over to you, Narendra Chirmule.
Great. Thank you so much again. So today I will speak about specifically the virology and immunology of the virus, but I'll touch on these points. And I'll talk about the now. What is now is so different. Every hour is different. Almost every minute is different, it seems. You open your computer or you look at the TV. Every day there's something new happening. I'll speak a little bit on the pathogenesis, meaning how the virus causes disease. I'll touch a little bit upon the treatment and where things are going. I've been fortunate to be part of a sub-team in India where we are having a discussion on the task force for advising the Department of Biotechnology and ICMR on various aspects of the COVID disease. I'll present to you the importance of diagnostics. Then we'll touch upon virology, immunology, and then because that is required to know what the vaccine would be. And then I'll touch upon a little bit of the roadmap of what might happen in the future. So obviously, I don't need to tell this audience or anywhere in the world what the numbers are going to be. It's a very important disease that we need to follow very, very closely. The virus is extremely infectious. Now, there are a lot of infectious viruses out there. This virus is actually not the most infectious virus in the world we have. The measles virus, the chickenpox, Ebola, other viruses are much more infectious than this virus. But one thing this virus is doing right now is it is infecting person to person, human to human. You don't need an intermediate animal like a mosquito for malaria to infect. You can actually infect the person directly, and that's what my dotted picture shows. And the dots in the green are the people who are not infected. And today, without a vaccine, without a drug, there is absolutely no way to protect against the infection other than social distancing. So this is the only way we can prevent the spread of this virus today. So a lot of people will ask the question, and me included, how long will the lockdown last? And today we've heard that the lockdown was extended till 3rd of May. And some very intelligent scientists have done a lot of modeling work to look at what's going to happen. So before we get into a picture on the left side, let's talk about what's happening in the 3 weeks. So in the 3 weeks, there will be those who get infected, and these people, they'll be healthy individuals. They should be able to clear the virus just like the flu virus. This is nothing like the flu virus, but the symptoms of the flu virus, and you'll clear it in 3 to 4 weeks. Then there'll be carriers who get infected, but they don't know they're infected because they don't have symptoms, but the virus in them will also be cleared. And then there'll be people who will be sick, and unfortunately, a fraction of them will die, and they will also be sequestered, obviously. And there will be majority of the people who are not infected. They are also sequestered because of the lockdown. That's what's happening in the 3 weeks. After the 3 weeks or the extended period that we are having till May now, the population will consist of majority of non-infected people. Then there'll be people who were infected and cleared the virus. Infected meaning either they were infected, got sick, or infected, did not have any symptoms. And the third group of people will be hospitalized and they will be sequestered. So we'll more about that and how we'll come back to normalcy later. And this is the extended period. So I'm going to address these 3 topics, which is for drugs, diagnostics, and vaccines. All of them relate to how fast can we come back out of the lockdown. How close are we to have a drug ready for coronavirus? Once we have a drug, how long will it take to control the virus? In the diagnostics, we have questions like, what will the diagnosis help? How will the diagnosis help control the disease? And what does the diagnostic test measure? And the vaccine, how far away from the vaccine are we? And will the vaccine protect? So let's look at the pathogenesis of this disease. And I've drawn a schematic diagram or picture to illustrate what is called the pathogenesis of the virus, which is how does the virus cause the clinical symptoms that we see. And the first thing that happens is the virus enters through what is called the nasopharyngeal route, through the nose, through the mouth, etc. And it enters the lungs. There, it infects the lungs, the lung tissue or the lung cells through a receptor called the ACE receptor, ACE2 receptor. The virus enters and it starts replicating, meaning it grows inside the cell. And this now, the virus buds out of these cells in the lung. And during the process, it kills the cell. So it's a cytopathic virus, meaning it kills the cell in which it grows. During this replicative process, when lots of viruses being made, these viruses induce the activation of an immune response, primarily the inflammatory response, which is characterized by proteins that are secreted by the immune system like IL-1 or IL-6. And these, what are called cytokines, are secreted by the cells of the immune system like the neutrophils, the macrophages, and dendritic cells. And the cytokines then spread all over the body, their inflammatory cells, and trying to find virally infected cells throughout the body. And because this ACE receptor is expressed on many different cell types, including the heart and the kidneys, etc., these inflammatory cytokines go and attack these other organs. So in the inflammation in the lung itself is caused both by the virus cytopathic effect as well as the inflammatory response in response to the virus, and you get difficulty in breathing. In the circulatory system, it goes to the heart, and then obviously the heart effects or cardiomyopathies are seen, meaning all the cardiac effects are seen, and you're more susceptible if you already are predisposed to some heart condition. And finally, it affects the brain, that you have fever, and then you have psychological effects, and finally you have multi-organ failure. And so therefore, the disease, and all of this happens in the course of literally days. It's a highly replicative virus, and that's what's the scary part of this disease, where from the time of infection till the multi-organ failure, it could actually just be a few days. So let's look at the size of the problem that we have at hand today in the world. I'm sure all of you are looking at numbers. I'm just going to put the chat on because in case there's a question. But there are a number of deaths that are occurring, and you can measure those. Then there's a larger group of people who are infected and treated. And these are healthy individuals in general, and they have the ability to be cured. Then there are a lot of people around them who are exposed and they're diagnosed. And so therefore they've been exposed, they don't have symptoms, but they've been diagnosed to be positive. Then there are even larger group of people who will be there who are exposed and undiagnosed because they don't have access to the diagnosis, or they don't have any symptoms and no reason to be diagnosed. And then you have the largest group of people, which is the entire population of the world, who need to be vaccinated to be protected against this virus, which is 7 billion people. So you can imagine the magnitude of the problem humanity has to solve this problem which we have at hand. So let's look at what is happening inside the body. So once the virus enters the cell in the lung, it's called an acute infection, the virus starts replicating. And this first period in which it replicates is what distinguishes it from many different viruses and organisms that infect humans, which is it has an incubation period, which is almost 4 to 7 days, where the virus is replicating rapidly, but you're not getting symptoms yet. So in this period, which is called the asymptomatic phase, there are a lot of people who have a lot of virus circulating in them. And if they are meeting anyone, talking to anyone, they will be transmitting the virus, but they don't know it. So this is probably the most dangerous group of people from a transmittability perspective. At one point, there will be a sufficient amount of virus which will then start causing symptoms, and these symptoms will be increasing, which peaks somewhere between 7 to 14 days. And then after that, the immune response is activated. And then ultimately, in about a month or so, the symptoms are gone. And they are gone primarily because the immune response kicks in. You first get a primary immune response, which is manifested by IgM antibodies, and then IgG antibodies, which is the secondary immune response, which clears the virus. So this is the course of infection and immunity in a normal, healthy individual who gets infected. In the symptomatic phase, while the first 1,000 deaths reported fever, cough, diarrhea was not common, and the incubation period was 4 days. As we've been hearing clinical symptoms from patients, we are seeing a much larger list of morbidities, which are listed here, and including things like loss of smell and taste, but ultimately pneumonia, psychological effects, pneumonia in the lungs, psychological effects of the brain, and organ failures result in death. So the manifestation of the disease is, one, is very rapid, and two, very difficult to reverse once it goes into this what is called the disseminated intravascular coagulopathy, which is the final stage of the inflammatory processes. So let's spend a little bit of time talking about the drugs that are being developed for this medication, for this disease. Since this is a new virus that has just emerged, there are no pre-existing drugs that are available. So many drugs that are already there are being tried to see. And the 4 top drugs which are being tested are being coordinated by a very large clinical trial called the Solidarity trial by the WHO. And they are testing 4 drugs. One is called remdesivir. The second we are all familiar with, chloroquine, hydrochloroquine. The third are anti-HIV drugs called lopinavir and ritonavir by themselves and with interferon beta. And what's interesting is that this clinical trial is a collaborative clinical trial with everybody in the world, and they can recruit patients into the context of this clinical trial. The results have not yet started coming. They will start coming in a few days, and we shall see which drugs will be more effective. All of these drugs have shown some efficacy in some parts of the world, and that's why a larger clinical trial needs to be done. One of the reasons so many trials are being done right now, and one of the popular sites that people go to look for which drugs are being developed, is clinicaltrials.gov. And you can look at these trials and see who's being recruited, where it is progressing. And the reason trials are required, because every drug has side effects. And we have to ensure that the side effects are not worse than the effect of the drug itself, especially in healthy individuals. Okay, so let's move on to diagnosis. I've come back to this picture that I showed you earlier, where I showed you acute infections, the viral replication, and then the immune response that occurs later. It is during this acute infections that you need a diagnostic to measure the amount of virus that might be present in an asymptomatic individual. And this is very critical because you need to be able to identify who is exposed to the virus. On the other side, you need to know that if a person was exposed, got infected, had symptoms, cleared the virus, whether that person is immune, and therefore you can measure antibodies to that virus so that a person who's antibody positive can be demonstrated to be positive, to have been exposed. The virus assay measures for the RNA in the virus. You're using a reaction called the polymerase chain reaction, and it is taken by a nasal swab. The sample is taken through a nasal swab. The RNA is extracted. Reaction mixtures are added to the reaction. The reaction is amplified, and then you detect. For antibodies, you look for what is called enzyme-linked immunosorbent assay. There are multiple forms. You take blood samples. A plate like this, a polystyrene plate like this, is coated with a viral protein. You add your serum samples. You amplify the reaction with a color reagent, and then you can detect who is positive and negative. So these are typical assays that are measured for viruses and antibodies. And there are a lot of different places where these methods are being developed. Now we come to vaccines. Typically, it takes about 10 to 20 years to develop a vaccine. And why is that? The reason is that you have to first identify which part of the organism you want to make the vaccine against. It's not as simple as just make the vaccine. You have to do a lot of research to find out which is the most protective antigen against which we can make a vaccine against. Then you need to express that antigen in a particular system, which is most optimal for that particular organism. Then you put an adjuvant, which is a stimulant of the immune system. You have to build a manufacturing process and a manufacturing facility to make this for a large amount of people. We said we're going to vaccinate 7 billion people. So this has to be a very large scalable effort. We need to find a correlate of protection. We don't know whether antibodies alone are sufficient to protect against this virus. There may be other parts of the immune system that might be required to develop a vaccine. And then finally, you have to do an animal proof of concept and then test it in clinical trials. And this is an iterative process. And that's why it takes a lot of time to figure out the best vaccine with the least side effects. More about the vaccine after we understand what the virus, receptor, and the protective immune response is. So let's look at what the virus is. Where did this virus come from, and why is it suddenly so infectious? So the coronavirus family of viruses are probably millions of years old, and they've come through various animal species, primarily the bat, feline, which is cat, canine viruses, which go into the alpha virus family. The beta virus family are defined by particular sequences, and this is the family in which the current coronavirus is part of. And it also infects camels, rabbits, not this virus, but the family of these viruses. And then gamma viruses and delta viruses infect avian and pigs. This coronavirus was sequenced, the genome of this virus was sequenced, and it is about 30,000 RNA bases, which is, as a comparator, HIV, which is also an RNA virus, is 9,000 bases. So this is at least 3 times bigger than an HIV virus. It has many proteins, which it encodes for. And all of these proteins, remember I told you, you need to find out which protein of the virus you need to make a vaccine for. It has more than 29 proteins. So which of these do you make a vaccine against is the challenge. So the origin of the virus, a lot of studies on sequencing has been done to identify where this virus may have originated. And by studying what is called the spike portion, which is one of the proteins on the surface of the virus, and looking at the sequence of that. And I'm just going to show you an enlarged version of this sequence portion that's shown here. And if you look at this sequence, which is showing here on the right, that little portion, this is the one portion of the spike protein. You can see CoV2 has an insertion of these particular amino acids. The BAT, the pangolin, CoV1, BAT2, and BAT3 sequences don't have that. So there's an insertion that happens right in that middle part of the spike, which somehow has made it much more infectious. And it is also highly glycosylated, meaning carbohydrate molecules can bind to this portion. So therefore, this spike protein is probably the reason, the mutation of spike protein is probably the reason why this virus has become more infectious. So let's look at the spike protein. What is the spike protein? This is the structure of the virus. It has these proteins called spike, which is a trimer, as you can see in this cartoon. It has the envelope protein. It has a hemagglutinin esterase protein, a membrane protein, and a nucleocapsid protein. And you can see in brown the RNA that it has. Digging deeper into the spike protein, the spike protein is made up of 2 sections called S1 and S2. And it has, in the between S1 and S2, there's a site, or a site meaning a sequence of amino acids, which is cleaved or cut like a scissors by a protease. Protease is an enzyme that cuts proteins. And so there's a site that has to be cut before it becomes infectious. And these are the list of different enzymes that have been implicated in cutting this spike protein for entry and fusion. This is a visual of the viral spike protein. And you can see that it has these two structures. It has this top portion, which is more of a circular portion, and the stalk. So therefore, for the virus to enter the cell, the top portion has to be removed or has to move before the stalk can bind to the virus and enter. So I've shown that in a small cartoon that I've drawn a little bit later. I'll show you that. The ribbon diagram structure of that looks like this. This is the top portion of the virus. It has that cleavage site and then you have the stalk portion of the virus. And this is the trimer structure of the virus that you can see, or the spike protein. In general, it looks like an umbrella, which is the top part is removed, and then you have the stalk. So let's look at how the virus might be entering the cell. And this is a cell, and it has two receptors, for example. A receptor is a part of the virus, a protein which is present on the surface of a cell, which the virus can specifically bind. This is a schematic of the virus with the S1, S2 portions of the spike protein. The spike protein binds to a particular receptor with very high specificity. Then what happens is the virus, the S1, S2 is cleaved, opening the other portion of the spike protein, and that's what allows the virus to enter. Let me just play that again. Virus binds to the receptor. It separates, because it doesn't completely separate, but it actually, that portion is cleaved, S1, S2 is cleaved, allowing the virus to enter the cell. So the entry process is quite complex. It is not a simple entry process. So not only does the vaccine have to be effective against this red portion of the virus, but it also has to be against the purple portion of the virus that's shown here. So the vaccine has to be it's not going to be easy to develop the vaccine. This is an electron micrograph of the virus showing the virus binding to a cell surface. In a paper published in Cell very early, and I have to actually make a comment on the scientific community before I go on. I must thank the scientific community for making all of these papers on this virus and the disease freely available to all scientists so that knowledge is shared across the world freely. I think that's very, very good. And congratulations to all the journalists and also the journals to allow this to happen. Thank you. So as I mentioned, back to this virus, the spike protein is a trimer. It contains 3 portions of the spike protein, and it forms this structure, which then forms this unit on the top, which is shown in red, which is the binding site of the virus to the ACE2 receptor, the receptor I showed you. So when this virus binds to that ACE2 receptor, it will infect the cell and enter, like I just showed you. What this paper demonstrated, that if you take serum or blood from patients who have been infected with COVID and have cleared the virus, which are called neutralizing antibodies, they are able to block the viral entry, demonstrating that an antibody against the spike protein may be sufficient to develop a vaccine. So that's very good news. Experiments done in days versus what it takes years to do. So what is the receptor? The receptor is a protein called the angiotensin converting enzyme 2, which is present on the cell surface of various cell types in our body. There's also a second receptor, which is called TMPRSS2. It is a serine protease, which is that scissor enzyme that I told you that needs to cut the spike protein for the virus to enter. So what is this ACE2? ACE2 is an enzyme which is required for controlling blood pressure and blood volume. And it is expressed on many, many tissues in our body and regulates this process by expression in lung, liver, kidney, adrenal glands, blood vessels, and many other cells of the body, which is shown in this picture. Scientists have done expression of this, levels of expression of ACE2 in our different organs, and you can see that almost every organ expresses ACE2. Colon has very high levels of ACE2, and you can see here is lung, you can see the heart, you can also see liver, pancreas, etc. And therefore, when this cell is expressed, when this receptor is expressed on so many cells, it is essentially allowing the cells to enter any cell in the body. And that is what overwhelms the system, because it is such an infectious virus. This life cycle of the virus is such: it enters the cell, it injects its RNA into the cell. While the RNA goes along and starts replicating, making new virus particles inside the cell, and killing the cell in the process. This is the virus is happening. In the meanwhile, the virus is also intelligent. It makes many other proteins. All those 29 proteins that are made by the virus also have a lot of functions. One of the functions is that, for example, these proteins listed here by the virus prevent the cell from killing the virus. So these are actually there. It's a very adept virus to attack the immune system. Okay, so what do we know so far? The spike protein of the virus is required for entry. Its trimeric structure is important. The spike protein enters through a binding and fusion process. The spike protein has been proven. It has been proven that serum from patients who have recovered can block entry. So we know that spike is required. So for developing a vaccine, what do we still need to know? We know what kind of vaccine will be required to express the trimeric structure. Remember, the trimeric structure is important for entry. What dose of the vaccine will be protective? And how often should the vaccine be given to develop these protective antibodies? How long will the protection last? And will the vaccine have side effects? All of these need to be tested in the context of a clinical trial. And this is the same picture I showed you earlier. And we hope, we meaning society at large, will hope that we will be able to develop a vaccine in 2 years, given everything that we know plays out in the context of the clinical trial. In the antigen identification, at least as of now, it appears that the spike protein antibodies may be sufficient to block entry. We shall see. The clinical trial will prove that. The expression systems can be many. It could be RNA, DNA, protein. It could be a viral vector. It could be inactivated virus or live attenuated viruses. And there are literally 40, 45 companies in the world who are trying to develop various kinds of vaccines to spike and to other proteins of the virus. And those are listed in the WHO website, where you can go to these websites and look at what's happening in the context of these clinical trials. The first vaccine trial started in the United States in March 2000, and therefore we can expect results pretty quickly to see whether at least there is some signs of the vaccine working. So one interesting concept in vaccine development is how do you test whether the vaccine is actually protecting against the virus? So one way that has been tried in the past is by giving the vaccine to people and challenging them with the live virus. So this sounds bizarre and really sometimes unethical. So the ethics community in science has debated on this topic and written a nice paper in Nature where they've demonstrated that this has been done before. It has been used from influenza, typhoid, cholera, and malaria. The advantage, of course, is to accelerate the approval process. And what does the process for this look like? First, you test to see whether the vaccine is safe. and that it induces the level of immune response that you have to show. You take a group of people who are at low risk from exposure, which are young and healthy people, give them either the vaccine or the placebo, wait for some time, and then expose them to the virus. There are obviously many risks. The risk can be reduced by giving it to younger people. That's what we understand today, that younger people are less clinically impacted than older people. And you select people from places which are endemic to the virus anyway. So those are the considerations of doing this challenge study. Whether a challenge study will be done with a vaccine or not is a question, but the ethics community has at least put out their position in this paper. So finally, after the vaccine is ready, what will society look like? It will have obviously the large number of people who are infected, and they will be sequestered or they will be in the hospital. So the infected people will be sequestered at this point. And then you will have a lot of people who have been exposed to the virus. They were asymptomatic, and they have resulted in antibody. They've created antibodies in themselves, and therefore they are prevented from infection again. And then finally, the people who are vaccinated will also be protected against the vaccine, against new infections. And that's what's called herd immunity, in which you don't need to vaccinate every single person on Earth. The green dots are the uninfected people. The blue are those who have been vaccinated, and the red are those who are infected. So you can see that if a person who is infected can transmit to the person who's not infected and not vaccinated, and this person will get infected. But then after this, it is impossible for this virus to infect the next person because that person is vaccinated or has antibodies. And this is what's called herd immunity. If there are a sufficient number of people who are vaccinated and or have antibodies, then you get an overall protection of society. And this is probably the scientific steady state that we will need to be before a full opening of the society happens. But that we'll have to see how the virus progresses over time. So I'm going to finish and just say that I spoke, when we talk about these kind of situations, we have to talk about these topics scientifically. And these are the things I try to cover: the natural history of the virus, the transmission and the diagnosis, what kind of research is being done for the discovery of vaccines, the epidemiological studies, which is how the virus is spreading, and that you get every day in various news items, the clinical presentation of the disease, prevention, control, therapies and vaccines, ethical considerations, and a lot of social discussion that is happening on this outbreak. So I thank you for your attention, and thank you, Atul and Shoolini, to give me this opportunity to present my thoughts. Thank you.
Thank you, Naren. What a fascinating talk. I definitely got goose pimples listening to it. I also understood a bit of the science behind the virus. I think lots of myths you broke, or you brought in new complications. But the question that still comes to my mind, which I think all of us want you to answer, of course, not from the social context, but from the scientific context. How far away are we when we can, without a doubt, walk on the streets? We can sit with people again, socially mingle with people, go to the cinemas, go to restaurants. What's your best guess?
It's a very difficult question to answer. Scientifically speaking, I think I'd shown you the picture of what we would, the kind of people the kind of population that we will have. If we have a vaccine, that will be easiest to control the spread of the virus, and therefore the development of the vaccine and vaccination will prevent the transmission, which we so take for granted for simple diseases like measles, mumps, rubella, hepatitis A, hepatitis B, which are also very infectious diseases. But since we get vaccinated, we don't blink an eye. But so a vaccine will be required to start protecting people from being infected. The other way, obviously, is to keep the people who are either have a good diagnosis. So if you, with the diagnosis, you should be able to detect people who are containing the virus, either symptomatic or asymptomatic. And then you can define people who are not symptomatic, which will be the majority of the people. If you have a diagnosis, simple diagnosis, and you demonstrate that you don't have either the virus or you don't have the virus, then you should be free to go into the society. So in the short term, I would say the diagnosis is very critical. And once we have a diagnosis, we should be all able to come out. And the vaccine will come shortly thereafter. And that's what I see that is required for normalcy to come back. Unfortunately, it's going to be a while before that happens.
Before we actually go into Q&A, I'll request you to stop the share screen, Naren.
Okay.
I think you're still sharing your screen at the moment. A few questions have come on YouTube, and I'll try to sort of summarize these questions. But the first, I think, big question a lot of the audience have is around, first, very interesting question, that if the antibodies which are created in the body, can we use the antibodies of recovered patients and use them for treatment? That's one. And of course, there's a general question which says that if someone develops immunity, do they develop immunity for the whole life? Is it for a year, multiple years? Would you like to pick this up, Naren?
Yes. Thank you for the question. Is my screen sharing off? I'm toggling. Is the screen sharing off?
We'll let you know. Why don't we start, Naren? We'll give you the feedback on that.
Okay. So yes, the answer to the first question is can you give serum from a patient who's recovered to another person? Yes, obviously you can, and it's being done right now. There are clinical trials to test that phenomenon. And it has been done in the past for vaccines, for viruses like Ebola, etc. It is not one of the safest mechanisms of giving antibodies from one person to another because you have a lot of other infections that can also be transmitted, unknown infections. But in extreme situations, I think it can be used, but I don't think it'll be genetically given to a lot of people. So that would be my comment. And the second question was, how long will the antibody last once you've been infected? I think what we know from virus immunology, like for example, for the chickenpox virus, etc., we know when you get infected with a particular organism, the protection of the the immune response develops a protective antibody against that virus. It lasts for at least 10 to 20 years. So it is likely that the antibody that is generated for this lasts for quite a long time, based on what we know from other viruses. But we'll see what happens with this particular virus.
Well, a very technical question from Adesh. I'm going to read it out. Can't make much out of it. So excuse me, all the big scientists out there. Is there any way to neutralize the spike protein of virus by a mimic of ACE2?
Good question. Very good question. So, yes, the answer is in principle, yes. But I would say from a development perspective, it would be easier to make an antibody or a vaccine against the spike protein rather than use the ACE receptor because of multiple things. One is that you'll have to dose the ACE receptor at a very high dose to be able to reach every cell in the body. You'll have to give it at a very high dose. So that's one challenge. The second is you don't know what the ACE receptor is going to do. That ACE protein actually binds to something on the cell. And one of the things that ACE inhibitors that you may be familiar with, which are given to heart patients, can actually upregulate, or meaning increase the expression of ACE on the cell. And so this feedback mechanism of ACE regulation has to be studied before something like this has to be given. But yeah, so that would be my answer.
Lovely. A couple of other questions. I think everyone is, there's a sort of a genetic feel we have that as Indians, we are more immune to the virus. Our immunity is superior. That's one question. What are your views on that? Then the question says that, it's not a natural virus, it's probably an engineered virus. So what are your thoughts on these two questions, Naren?
So let's go with the immunity first. So, there are a lot of hypotheses. So this hypothesis that people who are infected with or exposed to multiple infections and multiple bacteria and viruses and sort of dirty environments are more immune than people who are more sequestered and are not exposed to such pathogens. This hypothesis in immunology is called the hygiene hypothesis. And a lot of people are studying this very systematically, not just in the context of coronavirus, but also with a lot of different other viruses. So, I don't have a clear yes or no answer. Obviously, it's not black and white. It's not like people in countries where there are more infections that there is no coronavirus happening, right? It's happening everywhere. So it's not a black and white phenomena. Now, whether a hygiene hypothesis plays out and actually provides some higher level of immunity to people, time will tell. But my hypothesis would be it is possible, and the reason is because our immune system is needs to be educated all the time. So when our immune system is educated by either giving vaccines or exposed to different bacteria, you have a larger and repertoire of immune systems which are ready to which generate what are called memory cells, inflammatory responses, innate responses. These are more active, and therefore, if you have a more active immune system, in general, the probability that you will react a little bit faster to unknown pathogens is higher. That's a hypothesis that can be tested.
So before you get to the second question, Naren, this whole question about BCG, there's also a belief that as Indians, we've been vaccinated with BCG while the West hasn't. And that's another reason why you see less number of cases in India. Again, your scientific viewpoint on this.
Yeah, so good question. Again, there's a very nice paper that has been published recently about the epidemiology of people getting vaccinated versus not vaccinated. That paper has actually studied the number of infections in various parts of the world, like Italy and Germany and countries like that, versus other countries in which BCG vaccine is given as a policy of the company or of the country. And therefore, they see the statistical difference of the people who have got BCG vaccine or in our countries where BCG vaccine is given versus BCG vaccine is not given, and they see a statistical difference in the number of people who get infected in these countries. Now, while that seems to be the data that is shown from this paper, I don't think there's a specific study that has been done to demonstrate that a BCG vaccinated person is specifically protected against coronavirus. Again, it's an experiment not done, but it's a hypothesis taken from an extrapolation of some epidemiological data that is taken. Now, as an immunologist, I would say that, again, I would assume the same hypothesis, the hygiene hypothesis in the BCG case also, and that what BCG does, it actually does, when you get BCG vaccination, you do activate an immune response, which is quite strong. Mycobacterial diseases have elicit strong immune responses. So it's possible that this strong innate immune response may assist the anti-coronavirus immune response to become a little bit faster. It is possible. But why BCG? Why not any other vaccine? It's not clear. So unless somebody does a study in this systematic manner, it is difficult to speculate. Or it's easy to speculate, but it's difficult to prove. So that would be my answer for BCG.
So I think, second question, please.
Second question was whether it is a natural or a man-made virus. I think we can speculate and we can make movies about this and all that, but I think the reality is that, like I showed you in the sequence analysis, there was a very small insertion in the spike protein of this virus, and it is extremely related to the bat and other viruses of the same family. And these viruses, that's precisely how they mutate. When two different viruses, coronaviruses or two different species come together, there is some combination of genes that happens when they mix. And that's when a new virus is created. And therefore, the there's a high probability that this came from mixing of two viruses. I'll leave it at that.
So we'll just take two more questions. I think we're running out of time. A technical question by Dr. Shalu Sehgal. She does ask that, is there a significant difference between the spike proteins of type A, B, and C, nCoV?
I'm not familiar with A, B, and C, but are they different? Is the question, is the spike protein different from the different viruses, like the bat or other viruses, or is it within this COVID-2?
So I think, Shalu, why don't you get in touch with me separately? I think it's a technical question, and I'll have Naren answer that to you offline. But one last question over here. There's a belief that you spoke about BCG, you spoke about people who are more exposed to viruses, they're possibly more immune. But what about ethnicity? Are people of a particular ethnicity or a nationality more immune to the virus? What's your thought on that?
Yeah, so again, very good question and important to address. One can argue that certain countries have more infections than the others, et cetera, et cetera. Immunologically, susceptibility to viruses is dependent on the way your immune system is built. And these are regulated by, I'm going to get a little technical, molecules called major histocompatibility complex, which are basically the genetic material that you get from your mother and father. The immune response, the immune system that you develop has come from your parents. And depending on the genetic makeup of these immune systems, you can be more susceptible to one or the other viruses. Just as an example, there is a HLA, which is the MHA, major histocompatibility gene, which is called DR4, for example. You don't have to worry about the jargon. But if you have a DR4 gene of your immune system, your likelihood of getting rheumatoid arthritis is much higher than if you don't have that gene. So therefore, at least there are many, many diseases where susceptibility to a particular disease is dependent on the nature of your immune gene complex that you have. If that's true for rheumatoid arthritis, there is a possibility that could happen in humans also. I don't think it's race. It's dependent on this genomic sequence that you might have.
Great. I think what a wonderful discussion. Before we close, your 1 minute summary, Naren, 1 minute. What messages would you like to give us?
I think the most important message today for this virus is stay home and stay safe. Protect yourself. That's if you protect yourself and you prevent even a single infection from spreading, you're doing society a great good. I would actually end only with that, because that is the thing that we have to do right now. The vaccine is coming, diagnosis is coming, and drugs are coming. But we have to be patient.
Thank you, Naren. We know it's 3 a.m. in the morning for you. You still look so fresh. This must be, all the scientific work that you're doing. And it's also love, I guess. Love and tea that keeps you fresh. But once again, a big thank you. I'm definitely going back very educated, very excited, knowing that there are great people like you out there working towards a cure against the virus. Friends, thank you for participating. I think we had more than 300 to 400 people on the webinar today. We'll be back with you same time, 11:30 a.m. with another very interesting topic tomorrow. We'll be talking about the evolution of Hindi film music from ragas to Bollywood music. So if you have the time, Naren, would love to have you on that. By the way, before I close, apart from being a great scientist, Naren is also a great, he's really fond of classical music and plays the flute in an exceptional way. Probably next time, we'll have you play the flute live. Thank you, everyone. And have a great day.
Thank you so much. Thank you, Atul. Thank you so much.